Hello, today I will be introducing scDEED, a method for detecting dubious 2D single-cell embeddings. Hmm? Oh, is this better? Okay. Sorry, too short. Okay, thank you. So a common step in high-dimensional analysis is visualization, but it is well known that visualization tools like t-SNE and UMAP are sensitive to hyperparameter selection. So in this example, we apply t-SNE at 3 different perplexities, and all of the visualizations are different. And it's not just that they've been rotated, it's that the relative location of the clusters has totally changed in all the visualizations. So the question then becomes, which visualization should we use? And the answer to that question is probably the one that represents the structure of the original pre-embedding data the best. So current practices are that many people just use the default parameters. There are also some general guidelines, like the number of cells divided by 100 for perplexity, with similar modifications for the UMAP parameters. There are some dataset specific examples where researchers will try a variety of hyperparameters and select from the output visualizations. But the problem is that this is rather arbitrary and really depends on the user's pre-expectation of what the data should look like. There are some algorithms that attempt to optimize a loss function over a range of hyperparameter settings, but most of these methods are only designed for t-SNE and or UMAP, and so they can't be used to compare across dimension reduction methods, and they may not work for all methods. So we would like to introduce scDEED, a method whose purpose is to identify cells whose 2D embedding is visually misleading. And by that, I mean that the apparent 2D neighbors in the visualization are not representative of the neighbors in the pre-embedding space. Thus, the apparent location relative to other cells in the visualization is misleading, or what we'll call dubious. The identification of these dubious cells also allows us to suggest optimized hyperparameters by minimizing the number of dubious embeddings. The key idea behind scDEED is that the neighbors should be visually similar. So we capture this relationship by using distance vectors, with the distances calculated in the 2D embedding space between a cell and its nearest neighbors, with the nearest neighbors defined either the pre-embedding space or the post-embedding space. The pre-embedding space is usually the PCA space, but it's actually just whatever you would use as input to t-SNE or UMAP. So there can also be other ways besides just PCA. If the agreement is good for the distance vectors, like for this purple cell, then the correlation will be high, and this correlation is also the reliability score. For this orange cell, though, if we look at the pre-embedding neighbors and the post-embedding neighbors, their orientation is quite different, and so this correlation is going to be low. We can measure statistical significance under the null hypothesis that the neighbors after embedding are random. So to satisfy this null hypothesis, we can just permute the data. Once we permute the data, we remove all prior cell-to-cell relationships, so the neighbors are random. Then when we calculate the reliability scores, we can use this as a null distribution. By thresholding on this null distribution, we can categorize each cell as either dubious or trustworthy, depending on the cutoffs of 5th percentile and 5th percentile. And cells that don't fall in between those cutoffs are just unlabeled. So we will repeat this at each hyperparameter setting and then pick the hyperparameters that minimize the number of dubious embeddings to provide optimal hyperparameters. The other thing I want to point out is that we're only using the input and the output of dimension reduction methods. So even though the manuscript uses primarily t-SNE and UMAP, actually this framework can be applied to any dimension reduction method. As an example, we show the Hydra dataset. We have a reproduction of the embeddings from the original study with the author-provided annotations. We also applied scDEED to find that the optimized perplexity was 230. So at first glance, they're definitely different. If we look at the dubious embeddings, at the optimized perplexity, there's only a few dubious embeddings, and they're mostly scattered on the periphery of the clusters. But in the original embeddings, we have entire clusters that have been marked as dubious, which suggests that their cluster location is not reliable. And so as an example of what this means, we can focus on the neuron EC1 cells. So in the original embeddings, the neuron EC1 cells, which is one cell type, was split into two groups. So we have the red cluster up at the top that was marked as dubious, and then there was a second cluster that was marked as trustworthy, that was separated. And if we look at the gene expression between these two clusters, actually the gene expression is pretty similar. So this, combined with the fact that the authors did label this as only one cell type, really does suggest that there should not really be two clusters here. The separation into two clusters is not biologically meaningful, but just an artifact of the dimension reduction method. And if we look at the optimized perplexity of 230, we actually find that the neuron EC1 embeddings have been unified, which just further supports that the previous split into two clusters is not real, but just an artifact of the dimension reduction. We can also compare with the orange stem cell cluster, which has totally different gene expression than the neuron EC1 cluster. And this is reflected in the 2D visualization in which they're really far apart. And this is kind of what we would more expect, that distance in the 2D visualization should represent more different gene expression. So already this optimized perplexity is providing a more intuitive visualization than the original parameters. So the identification of dubious cell embeddings can help strengthen visual interpretations by highlighting cells whose gene expression is different than the cells around it. And this can be single cells or this can be entire clusters. We can also use these dubious cells to help optimize hyperparameters to create a more trustworthy visualization. Our manuscript is currently available in bioRxives and is under peer review at Nature Communications. We have more dataset examples. We also compare with some competing algorithms. And so finally, I'd really like to thank Lucy and Jessica for all their contributions for this work. Also Tianying for his work creating the scDEED package and other JSB lab members for their support. I'd also like to thank the NSF Mentorship Grant, UCLA Department of Statistics, and the BioVis Cozy Travel Fellowship, all of which allowed me to be here today. Thank you.
So, thank you. Questions?
Hi, thank you very much for this, I guess, amazing work, because it's a huge problem we're facing with single-cell RNA-seq data. My question is, do you think it is possible to have an explanation of what is it best, how do we choose between t-SNE and UMAP, for example? And is it possible to have like statistical tests to choose between both of the embeddings?
Yeah, so actually, because our loss function of just the correlation is independent of the t-SNE loss function and independent of the UMAP function, you could actually use scDEED to try both t-SNE and UMAP and see which one gives you a lower number of dubious cells. And we actually did try this for some data sets to compare across the two and found that if you change the hyperparameters, you can really reach the same number of dubious cells. But the amount of which you have to change the parameters from the default settings in many algorithms changes. So like t-SNE doesn't usually change as much, but as UMAP.
All right, one more quick question.
Could you comment on how scDEED works with batch correction efforts? And then also the example that you showed, was it done on only one sample, or it's a combination of multiple samples, replicates, and so on?
I think the Hydra one was only one sample, so that's the first one. And then the batch effect question, this one actually doesn't really work with batch effect, because the question of batch effect is how can I get two different samples into a joint embedding space? Whereas scDEED is saying, I have this high dimensional space. Now, how does it work when I put it in this low dimensional space? So if you had batch effect correction, you would need to first put your two different batches into the same joint high dimensional space, and then you could use scDEED to put that joint high dimensional space into the low dimensional space. So if you had like M&N correct, which just gives you the count matrix, the joint count matrix, or something like fast M&N, which gives you the joint PCA space, then that would work well for scDEED as a second step after that.